Hey everybody, and welcome back to the Sanctuary. I'm Professor C, and today we're going to be talking anatomy and physiology. Specifically today, we're going to do something a little different. We're going to look at a lab activity / review of all of the chemistry we've discussed so far. So, let's do it already. Alright, let's take a trip and grind some chemistry for a little bit. Make sure we can handle all the concepts from the previous lectures and apply them. And then, next time, we'll try an actual exam. So let's do some review. What do we got here? List the word name and symbol for 10 important elements. Okay, I can help you with this one. There is some trick I know. Imagine you live in a little town and your friend's coming to see you. And she's like, what are we going to do when we get there? And you're like, oh, we must go see Hopkins Cafe because it has the best food. So let me write that out. You must see Hopkins Cafe. And that is a very easy way to list the top 10 elements that make up living things. So I'm not going to write them out, but I'll say them out loud if you want to try them here. C for carbon, H is hydrogen, O, oxygen, P, phosphorus, K is potassium, a strange one, I is iodine, N, nitrogen, S for sulfur, Ca for calcium, and Fe for iron, which used to be called ferrium. That's why it's got the symbol Fe. So there's a good way to remember and list out very quickly the top 10 elements that are involved in most biological systems. Which four make up most life? Well, we know this answer too is just CHON. Carbon, hydrogen, oxygen, nitrogen. List the 3 subatomic particles, their mass and their charge. Well, that would be a proton, a neutron, and an electron. Subatomic particles. Proton is plus, has a positive charge, and we know they weigh 1 AMU, if we want to get fancy. Neutron has no charge, it's neutral, and it also weighs 1 AMU. Electron has a negative charge and weighs nothing according to our math. All right, no problem there. Given the following information, draw Bohr models for each atom and fill in the empty cell. So this might take a little bit of work to do. Let's start with the chart real quick, and let's fill out as many cells as we can without knowing anything. Let's start at the bottom. Well, I know protons and electrons should be the same. When I'm drawing a Bohr model, I should have the same number. So in atom C, it says it has 8 protons, so we can assume it's got 8 electrons also. That we can figure that one out. Same thing with atom D. It's got 6 protons, so it's probably got 6 electrons as well. All right. Well, now we can completely figure out the rest of C, can't we? We know to get an atomic number, we just need the number of protons, that's 8. It's given to us. And the atomic mass, or atomic weight, is just the neutrons and the protons together. And so neutrons and protons together is 16. All right, what else can we figure out here? Well, atom D. Again, we've been given the number of protons, so the atomic number is the number of protons, 6. And we've been given atomic mass. We know that the atomic mass or weight is the protons plus the neutrons. So if 12 is the mass, and we've got 6 protons, well, we must need 6 neutrons to equal 12 weight. A lot of 6's there, a lot of 8's on the top. Okay, fine. Let's see what else we can figure out here. Atomic number has been given for atom A and B. So atom A says atomic number 10, must mean it has 10 protons. Also means it's got 10 electrons because they're going to be balanced. Atom B has atomic number of 2. By definition, that's 2 protons, but it will be balanced with the number of electrons and have 2 also. And then we can use the trick that we used on atom D and work backwards. We've been given the atomic mass on atom A of 21. And we know that is derived by the weight of the protons and the neutrons. So there are 10 protons. The atomic mass is 21, so I'm missing 11 neutrons. They don't have to be all the same number. Okay, atom B. Same game, we've been given an atomic mass of 5, right? And we know that's the weight of the protons and the neutrons, and there's 2 protons, so there's 3 left. This should be the number of neutrons. And if you want to sketch that out real quick, let's do it. Atom A, 10 protons and 10 neutrons, 10p, 10n. Electrons are 10, so you'd have 2 here, and you need 8 more. Something like that. And you're done with atom A. Atom B, looks like a tiny one. 2 protons, so we put 2p and 3n in this case. We derived that. There's going to be 2 electrons, and that's going to fill up that first shell, and we're done with that one. Atom C, it has 8 protons and 8 neutrons there in the nucleus, which means it's going to have 8 electrons also, so there'll be 2 in this first shell. And then 6 left. Can I put them all out here? I sure can, because I can hold 8. Just like that. Okay, and then atom D, 6 protons and 6 neutrons in the nucleus. That means I need 6 electrons. 2 will go in the first shell, and the other 4 will fit out here. And we did it. We filled out the chart by a little bit of logic, and we drew the Bohr models just like we practiced in lecture. Okay, based on the following info here, determine if these atoms, A, B, C, D, E, are charged. Big charge, right? Not just an electrostatic charge. And label which ones are cations or anions. Okay. Atom A, I've got 11 protons. That's 11 pluses. Remember, if you need to help yourself, make a little note on your test if you can. So 11 plus and 10 minuses. Well, who wins? Well, overall, the pluses win. And this is a cation. All right. Atom B has 17 pluses and 18 minuses, so it's got 1 extra minus than the plus, so 1 extra electron means it's going to have a charge of -1. That would make it an anion. Atom C has 2 protons, 2 pluses, and 1 electron, 1 minus. That gives us an overall charge of +1, and again that would be a cation because it's positively charged. Atom D, 11 and 11. Okay. 11 protons and 11 electrons, that's balanced. So the overall charge is nothing. You would not call it an ion. That's just a neutral atom. And then atom E here, 20 protons, 18 electrons. There's 2 more protons and electrons, so this would have a +2 charge. It's not a double cation. It's just called a cation because it's positively charged atom. There's how to deal with that. Now, if the question were, which of these 2 atoms are isotopes of one another? Let's change it up a little bit. So, which are, that's a different question, which are isotopes of one another? Well, what do we look for when we look for isotopes? We look for same atomic number, which is same number of protons. So same, let me change my color here. I'll write it right here. Same, we were looking for same protons, but different neutron number. And the only place I see that is atom A and atom D. Right? This has 11 protons, that's the same atomic number, but they differ in the amount of neutrons. So A and D are isotopes of one another. Define anabolism and catabolism. So anabolism is a building, and a catabolism is a breaking of bonds. And that's about as simple as you can keep it right there. Anabolism builds and catabolism breaks. Show an equation for synthesis, decomp, and an exchange, and any alternative names, and then apply the terms endergonic, exergonic. Okay, let's give it a shot. Synthesis reaction, let's start with that one first. It would be A plus B merging together into a single thing. So there's a synthesis. I'll just put that. Synthesis. We know if we create a bond, we have to put energy into the system. So that would be an endergonic reaction. You also might remember we have to pull water out of the system to make that work. So we could actually call that a dehydration reaction as well. All right, let's look at the next one. A decomp is the opposite. We start with AB, and it will be broken apart into A plus B. So a decomposition reaction. We know to do this, you have to add water to get that bond to break. You can call this a hydrolysis reaction because we're adding water to break a bond, and energy will come out of here because we've broken the bond, so this would be an exergonic reaction. Now an exchange is something a little bit different. It would have like AB plus C, and then one of them would break apart, right, and hook up with the other one and leave the other original partner all alone. You could call this displacement reaction. And as far as endergonic, exergonic, that doesn't really apply here. We are both making and breaking bonds at the same time. List 4 factors that cause the rate of a chemical reaction to change. Well, that's very easy. Temperature, pressure, pH, and enzymes. There are others too. You could talk about the concentration of the reactants and other things as well. But usually you hear temperature, pressure, pH, and the influence of enzymes. Explain the difference between polar and nonpolar covalent bonds. Very simple. Nonpolar is equal sharing, and polar is unequal sharing. And that's the simplest answer there is. Write the structural formula for water. Okay, H2O. Got that done. And indicate polarity. All right, remember we had a big O, and we had the two little H's out like this, and the molecule was bent at an angle like that, right? Because this giant electronegative oxygen was pulling so hard on those electrons that it bent the molecule in space. Show how water molecules orient with one another. Okay, well, we know that we could have another water molecule here, like so, and this giant electronegative atom, let me change the color so we see that better. This giant electronegative O and this giant electronegative O are both tugging on this poor little hydrogen. And that makes an H bond right here. Show how water molecules orient with one another, like that. Show how a water molecule would orient around a cation and an anion. Okay, something like this. We could have a cation, like sodium, and you would find the oxygen portion of the water molecule, the negative part, around it, like so. However, if you had like a chloride ion, I'll draw it over here, you would have the H's, the hydrogens, would be associated with it because it is the positive part of that polar molecule. All right, what do salts, acids, and bases have in common? They both ionize or dissociate in a watery solution, and they conduct electricity. They're electrolytes is the simplest answer. So they are electrolytes. That is, that they will dissociate in a watery solution and then conduct electricity. And you see that at the bottom. What is the phrase, potassium chloride ionizes in solution? Well, let's look at that. Potassium is K, chloride is L. We squish them together in an ionic bond. Potassium chloride ionizes in solution. So that means, I don't know what that line came from, but hey, We put it in water. We put it in a watery soup. Take some potassium chloride, a salt, and put it in water. What happens? Well, it goes into its ionic components. Potassium ions and chloride ions, very similarly to the way we saw NaCl, or table salt, NaCl, sodium chloride, do its thing. So there is potassium chloride ionizing or dissociating in solution. All right, if the pH of a system changes from 3 to 5, what is the change in hydrogen ion concentration? Okay, so remember this. Let's help ourselves out. pH scale goes from 0 to 14. 7 is in the middle. This is saying pick 3 and 5, which is, you remember, on the acidic side, and this was on the alkaline side. So 3 to 5, it's changing from 3 to 5, it's going this way, and remember to count the steps. 3 to 4 to 5, there's 2 steps there. 1 step, 2 steps. And what we do is, we multiply by 10 for each step. So the pH of a system changes from 3 to 5, we're going to have 100 times fewer hydrogen ions because we're going up toward the 14. This one down here at the bottom left corner, it says how about from 7 to 10? Well, we have 7 already listed. 10 would be somewhere about here. Again, count the steps. 7 to 8 to 9 to 10, where we want to get to. There's 3 steps. So you put a 1 and then you put 3 zeros 1,000 times. So we're going to say if you're going from 7 to 10, what's the hydrogen ion concentration change? It's 1,000 times fewer again. Fewer because we're heading up toward the higher numbers. 1,000 times fewer. Let's do another one more at the bottom. So 3 is here. 2 is here. We're going this way. It's only 1 step, so it's very simple. We just put a 1 with 1 0 10 times, and is it more or less? Well, we're headed toward the 0 now. We're headed toward the acid side, so it's more. A pH 2 would have 10 times more than a pH 3, more hydrogen ions. List the 4 major classes. Well, this is easy to do. We have carbs, lipids, proteins, and nucleic acids. And you could say carbs is, some kind of starch used for energy storage. You can talk about lipids being fats and oils used for energy storage and insulation and protection. You talk about proteins functionally as structures in a cell, as antibodies, as receptors, and many, many, many others. And then nucleic acids would be DNA and RNA, and they would function in some sort of information carrying in a cell, going from pure raw information into how to cook up a protein. List the subunits of the following organic molecules. Okay, fine. A starch is a carb. Therefore, it's going to be made of, well, it's not only a carb, but it's a polysaccharide. It's going to be made of individual monosaccharides, and that's it. Monosaccharide. Glucose, to be specific. A protein is a string of what? Amino acids. So I'll just kind of cheat and put AAs. Protein breaks down into amino acids. Triglyceride is a glycerol. And it's got a glycerol head plus 3 fatty acid tails, 3 caterpillars. I'll just put 3FA. Phospholipid is similar to a triglyceride, but it had a head and only 2 tails. And then a special phosphate head stuck on top. So it's made of a glycerol head with a phosphate head and 2 fatty acid tails. And then DNA is simply made of nucleotides. All right, so there are the building blocks of these major organic molecules. Okay, 2 pentoses, very simply deoxyribose and plain old ribose. And then hexoses were 6-carbon monosaccharides. That would be glucose, fructose, and galactose. Yeah, there are others, but those are the ones you see on almost every exam. List 3 disaccharides and name the 2 monomers that make each. So glucose, this is a straight memorization game. Glucose plus fructose equals sucrose. Glucose plus galactose equals lactose. And then glucose plus more glucose equals maltose. All right, list 3 polysaccharides and give their functions. Well, we talked about plant starch, which was some way for a plant to store sugar. We talked about animal starch, also known as glycogen, which you store in your liver and muscles, store energy, a way to store energy. We talked about cellulose being a long chain of monosaccharides, and they functionally act as a structural fiber, like plant fiber. And we also mentioned chitin, and there are, of course, many others, but these are the ones you usually see: starch, glycogen, cellulose, and possibly chitin. How do proteins form? Show the levels of protein folding. Well, very simply, first order, or primary, is just the sequence of amino acids linked by peptide bonds. If we wanted to go second order, we could either make an alpha helix, or we could turn it into some sort of pleated sheet structure. So there's our 2 options, an alpha helix or a beta pleated sheet. At the third level, or the tertiary level, we'll have a globular protein, and we'll take, say, an alpha helix and a beta pleated sheet linked to a helix, linked to a pleated sheet, and you'll start to get this 3-dimensional globular protein form. And of course, take many of those, and I can show that just by changing the color. Take another globular protein here and link it up like that. Take, say, a third globular protein and hook it up like this. And now we have the quaternary, or the 4th level of protein folding. And then denaturing is just when you start to unravel any of these structures. If any of these structures lose their form, especially that 4th or 3rd, then you lose your structure and you lose your function. What is the building block of nucleic acid? It's a nucleotide. Show and label its components. Remember, it's got 3 parts. It's got a phosphate group, a pentose sugar, which could be either deoxyribose or ribose, and then hanging off of the sugar is some sort of nitrogenous or nitrogen-containing base. Could be guanine, adenine, cytosine, thymine, or possibly uracil, depending on the nucleic acid we're talking about. Compare and contrast DNA. Well, to keep that real simple, we can do something like that. Number of strands, 2. RNA has 1. We could talk about bases. We know DNA has G, A, T, and C, guanine, adenine, thymine, cytosine, and RNA has G, A, U, C, uracil, instead of thymine. Sugar, the sugar in the backbone. In the D, well, it's going to be D is the name of the sugar, deoxyribose. And the name of the sugar in RNA is simply ribose. You could talk about function. DNA is information, how to create proteins. It's located in the nucleus. And RNA is more of a worker, doing the orders that the DNA has instructed it to do, and will help build up the actual protein. All right, that was a pretty good study session. Next time we'll do a full test, and we'll see if you can handle some multiple choice questions. Until then, bye-bye.